There we go. We are now being recorded. Sweet. So chapter 7, this is going to be the amino acid, nitrogen, and urea cycle. Basically, what this whole chapter is about is with our amino acids, like the whole chapter before this, we were breaking down amino acids, either like to make ketone bodies, to make glucose, to make like the glycolysis or the Krebs cycle intermediates, or breaking them down, to use their parts for whatever that we need to use them for. We're left over with certain parts of our amino acids, specifically the nitrogen, that we need to be able to get rid of. We have to do something with that nitrogen in order for our bodies, to be happy. Because this NH3 here, so there's you'll see a couple different forms. You'll see NH2. Let me actually type these because it'll look nicer. There's NH2, which is an amino group. NH3, which is, I have it written down here. It was the ammonia, and then NH4 is the ammonium. They all kind of essentially mean the same thing. Like they turn into one another, basically. But sometimes, like if you see the different names, might be a little confusing. So NH2 is the amino, NH3 is ammonia, and NH4 is ammonium, just in case he uses any of those different words. Our first question: which of the following amino acids is/are produced in the urea cycle? So we look specifically at the urea cycle. Some major components that we're going to be looking at with the urea cycle are going to be like the liver, the kidneys, the muscles, all those kind of places. I'm going to find, there was a nice picture here of the urea cycle. Here we go. So we can look through here. Unfortunately, again, this is just another one that we need to draw out, kind of memorize, figure out what the heck is going where and what turns into what, because that's just part of biochemistry. But we start up here with citrulline, which if we take a quick step back, there's another kind of you can kind of compare this a lot to, it looks very similar kind of to like glycolysis and then Krebs cycle. It's kind of similar to that in the fact that there's like these two separate little reactions. We start with our NH4, that ammonium, and we have this carbamoyl phosphate synthetase 1. This is known as CPS1, and that guy is going to convert our ammonium into carbamoyl phosphate, and then we combine it with ornithine to turn it into citrulline. Essentially what we're doing was taking this NH4 and we're plugging it in onto like the carbamoyl or the carbamoyl phosphate to turn it into this, and in order to prep it to enter into the actual urea cycle. So here we have the ornithine, citrulline, same thing there, ornithine, citrulline. So that happens, when that first step happens in the mitochondrial matrix, then the citrulline comes out of the mitochondria into the cytosol, and we start our kind of like our urea Krebs cycle, as well. I'll put that in air quotes. Citrulline gets acted on by the argininosuccinate synthetase, gets converted to the citrulyl AMP intermediate, keeps going, it turns into arginosuccinate. Argininosuccinate synthetase makes arginosuccinate. Arginosuccinase then acts on it to turn it to fumarate, where you get fumarate and you get arginine. Arginine continues arginase to ornithine, and that ornithine goes back to the mitochondrial matrix to keep going around. So parts you can kind of, studying this and reviewing this, I would kind of cut out this part right here. Don't worry too much about that. You're going to want to know the enzymes. Luckily, the enzymes are pretty similar to the things that they act on. Arginine, arginase, argininosuccinate, argininosuccinase, argininosuccinate synthetase, all kind of goes along there. We look at the question though, in the urea cycle, which are produced in the urea cycle. Citrulline we saw is the first step. Arginine is a little bit later on, and then ornithine is that last step. So this answer is all of the above. Okay. Next question. Blank is an allosteric activator of carbamoyl phosphate synthetase 1, that's CPS1, in the urea cycle. Liz, do you remember what allosteric activator means?
Allosteric inhibitors are going to bind to the site. So allosteric activator, is that the where basically the substrate binds where it's supposed to?
So allosteric is going to be where it doesn't bind to the actual active site.
Okay.
So allosteric means, so it's of, relating to, or being a change in the shape and activity of a protein that results from combination with another substance at a point other than the chemically active site. So the spot somewhere else besides like the specific receptor site. So something comes and binds, which changes the shape of the enzyme slash protein, binds somewhere besides the active site, which changes the shape of the enzyme protein. And in this case, because it's an activator, it activates it. The way that I always remember this is the carbamoyl phosphate synthetase 1, the CPS. Kind of a stupid way of remembering it. The CPS, a lot of times they deal with complaints, they deal with nagging. So the N-acetyl glutamate, that's going to be the name of our one of our allosteric activators that deals with it. So you nag the CPS. Nag the CPS. Boom. Hello, Kristen. Welcome to tutoring.
Hello, I am driving, but I am listening.
Sounds good. Okay. Next question. The amino acid catabolism involves the breakdown of 20 amino acids, all of which contain nitrogen, but have different carbon skeleton. What overall strategy is used to deal with this problem? So we look at this, if we actually go to the book and we look and see kind of the fate of this. So the amino acids are engaged in synthesis, like fatty acids, lipids, carbohydrates, neurotransmitters, all those kind of stuff. The extra ones get broken down, and we'll get the carbon skeleton and the nitrogen skeleton. So we have, you know, the carbon skeleton is kind of this whole part right here, actually including that. And then the other side is our nitrogen skeleton. Nitrogen skeleton, carbon skeleton. So the carbon skeleton specifically is going to be used for essentially for the gluconeogenesis part of things, right? Because we have two carbons like our acetyl CoA, a lot of our intermediates within the Krebs cycle have the kind of that two carbon base, and also like the R group will typically have additional carbons. So this whole chunk will kind of go off. And if it's a glucogenic amino acid, which we talked about last week, you have the different glucogenic amino acids versus the ketogenic amino acids. The ketogenic are the leucine lies to get the keys, that leucine, lysine, mainly for that focus, compared to the glucogenic ones, which can go into the Krebs cycle. So that the nitrogen skeleton, we're going to have a way to get rid of, thus the urea cycle. The carbon skeleton we're going to use to make intermediates. So when we look through this, these are the answer choices. Personally, the way that this question is asked, it looks like it's more focused on the carbon skeletons. I think a better answer would be the carbon skeletons are used in gluconeogenesis or ketone production. That's a potential answer. And honestly, if that's the answer that was available, that's the one that I would like. But we look at the rest of these, the carbon skeleton is converted to form an alpha keto acid. That's not quite the case. And then these other ones, we could dive deeper into them, but right now I don't think it's the best use of our time. But at the end of the day, so for this specific question, this is going to be our best answer. That the nitrogen is removed by transamination to glutamate. There's another word that we've used before, transamination. Do you remember that word at all? Kind of those transferase type enzymes. So transamination.
I remember that we've talked about it, but I don't really remember it.
Yeah. It's literally, it's a chemical reaction, so it's a type of enzyme. Our transferase enzymes transfers an amino group from an amino acid to a keto acid, which creates new amino acids and oxo acids. That last part's not super important. This first part is the one that you're going to get questions on. Is that the transamination is a chemical reaction that transfers an amino group from an amino acid to a keto acid. So essentially, because what we want to do is pass along this that nitrogen to give us our carbon skeleton or a keto acid to then be able to go off and do what we need with it. But we have to get rid of that nitrogen somehow. The best way to do that is through this transamination reaction, where we use the aminotransferase enzyme to remove it off of one amino acid. We stick it onto an alpha ketoglutarate. Alpha ketoglutarate is typically coming kind of from that Krebs cycle that's going on. And when we stick on that nitrogen, it turns into glutamate. So essentially we're forming a bunch of glutamate, and we'll see why kind of with glutamate, there's another kind of shuttle type system that helps us manage the glutamate and stuff that we make, and then kind of where exactly to send it. The important part, another important thing to remember, so two big things from here. Let's say three. First one is amino acids get turned into alpha keto acids by removing their nitrogen group. The enzyme that does this is the aminotransferase, and it turns alpha ketoglutarate into glutamate. And we use a cofactor that is pyridoxal phosphate. Pyridoxal phosphate is one of our B6 enzymes. I think it was technically 4 things, but those are the big things to remember from here. If you're going to bind a lot of the stuff here, you're going to see, guess kind of glutamate or guess alpha ketoglutarate. Those are going to guess glutamate first. If that's not available, alpha ketoglutarate is probably a good answer choice as well, because those two both pop up pretty frequently in this chapter. Yeah. Certain diseases, such as hepatitis B infection, can lead to functional liver cell damage, interfering with amino acid metabolism, and can produce coma. Which of the following reasons best describes the development of this coma? So if the liver is having issues, look at a couple of these answer choices. It's a big bug. Inability to metabolize glucose. Obviously, we have other parts just besides the liver that can metabolize glucose. The glutamine synthase activity, that has to do with a whole other process that we'll look at as well. Overproduction of aspartic acid. Aspartic acid is not going to be being produced in the liver. So the ammonia intoxication, because one of the most important jobs of the liver is kind of that cleaning detoxification. Detoxifies blood. So without the liver, we don't clean out the ammonia. And that is bad. We also think ammonia, right, when we looked at that, that has a fair amount of hydrogens. And if we put ammonia into something, a lot of hydrogen, it is going to act as an electron, like as a hydrogen donor. It's going to act as an acidic. If we start releasing a bunch of hydrogen and stuff into the body, that messes with our pH. Messing with the pH can have a whole bunch of big old negative effects, which in this case, for example, can produce a coma. We'll look at this again a little bit later, but I'm sure in these questions it'll ask it. But the two big structures that we look at with kind of part of the urea cycle are the liver and the muscles. There's a lot of kind of communication and transportation of stuff between the liver and the muscles, which we'll get to. Okay, conversion of arginosuccinate to arginine is a step in which of the following? This one we just saw, we looked at it. We have the urea cycle. With the urea cycle, we start off the very, very beginning, before like the actual loop part of it. Where does this process begin? Where does the urea cycle, like pre-process, begin? In what, inside of what structure?
The matrix, mitochondrial matrix.
Yeah, good. We begin inside of the mitochondrial matrix. From there, I'm going to take a look at a beautiful little picture. We have our carbamoyl, we have our ammonia, this CPS1. Do you guys remember what was the allosteric activator of CPS1?
Alanine.
Remember the abbreviation of it?
It's the NAG.
Good, the NAG, because you nag the CPS. So it's N-acetyl-glutamate. Again, glutamate's going to keep popping up. NAG, N-acetyl-glutamate, is that activator. So we look at our full process. The CPS turns us into carbamoyl phosphate. Ornithine comes on over, picks up that H2N, everything kind of transfers over, turns into citrulline. Citrulline goes to cytosol, citrulline to arginosuccinate, arginosuccinate to arginine, arginine to ornithine, and then back around. It goes and goes and goes. And here's where our urea kind of pops off. This is what you'll want to remember, especially that from arginine to ornithine, thanks to the arginase enzyme, that's when we actually get the urea. Occasionally, that's a question that he'll hit on. Okay. During a transamination reaction, glutamate that is present in the skeletal muscle, here we're coming back, we're talking about skeletal muscle, that's present in skeletal muscle tissue transfers its amino group to another compound. Thus, a new compound is produced within the muscle tissue. This newly formed compound is then transported into the blood circulation and finally enters into the liver tissue. Identify the compound to which glutamate would transfer its amino acid or its amino group during this transamination reaction in muscle tissue. There's a really good picture that Sarkar has in his slides that I think does the best job of this. This picture right here is the one that I really like. So amino acids, if we think way back to Biochem 1, the way that we produce proteins, that we build up protein, that we build up muscle, that we make all of our stuff is through taking amino acids, putting them all together into a peptide chain, and then folding it all nicely to produce proteins. We know the muscle is made up of a bunch of these protein type structures, everything that's going on in there. So what happens is there's one other picture as well. Let's see if I can find. There's one that's even simpler than that one, actually. Pretty sure it is in this slideshow.
It's in this week's, or the slideshow he had last week for our presentation, I know.
Yeah.
That one right.
Yeah, this one right here. So the amino acids in the muscle tissue, we have glutamate. Glutamate is going to pass that nitrogen to an alanine. So glutamate passes it along to alanine. Then the alanine I want to make sure but I want to make sure this question might have an incorrect answer choice as so just as we see it. Okay, here we go. I think this question is worded a little bit incorrectly. We'll look at the whole process and then. So essentially, what you want to know. What's it? Okay. So the amino acids in the muscle tissues get broken down, and then we get the ammonium that's here, and that's bound onto the glutamate. That glutamate, like I said, this is the good picture. Glutamate in the muscle gets passed on to the pyruvate. When it gets passed on to pyruvate, that converts into alanine. Alanine is what travels into the blood, makes it to the liver, turns back into the glutamate, and releases pyruvate. Pyruvate goes through gluconeogenesis, turns back into glucose, and comes back up as glucose back into the muscle tissue. The big key things in the way that he's going to ask these types of questions most of the time is going to be, hey, which of the following, mostly which is traveling in the blood. One sec. Hey. Okay. The key things that are going to be kind of the focus is what is coming from where. So the alanine comes from skeletal muscle. So from skeletal muscle, we get the alanine. And the alanine is what travels through the blood. And then that goes to the liver. The liver turns it back to glucose. And in that process, urea pops out. And that glucose goes back to the skeletal muscle to be used by the skeletal muscle. And the process repeats over and over and over again. So that's the general idea, the one that you're going to really want to know. This question, he'll probably ask maybe this specific type of specific question, in which case, in this case, the answer is pyruvate. I don't love the way that this is worded because technically with the compound to which glutamate would transfer its amino group during the transamination reaction in muscle tissue, if we look at a lot of this, technically glutamate isn't it really isn't as super involved. Glutamate is more kind of seen on the other side of things, but it's fine. It's a glutamate. Here in muscle tissue, glutamate hands it to pyruvate to turn it to alanine. On the opposite side of things, we could rearrange this question to say, identify the compound to which alanine would transfer its amino group during this transamination process. Alanine would then transfer it to glutamate. Sorry, a little bit confusing there, but that whole this little loop, knowing the generals, kind of what is where, very, very going to be very important for this exam. And I remember there's a lot of more lengthy questions in this kind of review. I do remember him having quite a few questions on his exam that were a little bit more simple, a little bit more straightforward. This one probably has a lot of words, but idea-wise, it should be pretty simple. So during a transamination reaction, glutamate present in skeletal muscle tissue transfers amino acid to another compound. It transfers it to pyruvate. New compound. This newly formed compound is then transported into the blood circulation. That newly formed alanine is what's traveling in the blood and finally enters the liver tissue. Identify the new compound formed, which is transported into the blood circulation. So a lot of words, but which one is in the blood circulation? Our alanine is in the blood circulation. It was a little bit confusing there for a sec. Any specific questions on this little process?
Good here.
I think I'm good here.
Okie dokie. During amino acid metabolism, glutamate releases its blank group as blank in the blank. This is where we start to look at those things. So glutamate coming off of the amino acid. The amino acid itself, we look at the stages. When the amino group is on the amino acid, it is our NH2, which is an amino. Immediately we look for when it's coming directly off of an amino acid, it's an amino. So look here, immediately these two are gone. It's one of these two. And we look, so glutamate releases its amino group as blank in the blank. So if we look at this, we think N2, all of our amino acids only have one nitrogen, so it can't be that. And so it's going to be A. Amino releases its amino, so glutamate releases its amino group as ammonia, so it picks up another hydrogen, NH3, so NH2, NH3, and that happens inside of the liver. We look back at this one. glutamate right here, releasing everything. Or did it come from urea inside of the liver? I say this on the recorded video of this chapter. Not an expert in biochemistry. I do think some of these graphs and charts, because so many different people make them, they're slightly flawed or like slightly different, or like little steps are skipped. So they're not all perfect. It's fine as long as we get the main idea. Glutamate produced within other tissues must be transported to blank for urea production. So where is our urea production going to be? Where did we say? Where is that going to take place?
The liver?
Yeah, the liver. The urea production happens in the liver. So we'll say liver is production. Skeletal muscle, we always associate alanine that goes to the blood with skeletal muscle. And kidneys, when we talk about kidneys will filter out the urea. And the brain, what happens if we don't get rid of all of our urea in our body and it starts to build up, like if the liver is having issues?
We get really sick.
Yeah. A lot of times we'll talk about going into a coma. Do one more question, then I gotta run to help put my baby to bed really quick. Okay, so glutamate produced within other tissues must be transported to the liver in the form of which of the following? The transported to the liver, it's coming from our skeletal muscle. It's always going to be, yeah, our alanine. So alanine is the one. It's always coming in as alanine. He likes this question. Okay, will be back as soon as I can.
Sounds good.
I'm back. Let me know if you guys are back or if you ever left.
I mean, I never left. I just kind of had it on mute. I like my computer.
I'm back. I was just calculating grades to see what I need to make to pass.
Perfect. That's a slippery slope. Be very careful with that one. Okay. Next question. How many amino groups are present in urea? This is what our urea molecule looks like. H2, NC, and H2O. So we have 2 nitrogens per urea molecule. That's honestly just a, hey, you need to know that, what it looks like. We get 1 ammonia from each, glutamate, this 2 go into urea. Okay. The urea cycle ornithine reacts with which of the following to synthesize citrulline? Just think up there at the very, very top of that reaction. And we start out with our ammonia. Ammonia messes with, we get our CPS 1 that's activated by that NAG. We have B6 that's active in there as well. There's like some ATP and some other stuff messing with there. We get carbamoyl phosphates. And then our ornithine comes and interacts with our carbamoyl phosphate to give us our citrulline. So in this case, the urea cycle ornithine reacts with the carbamoyl phosphate, and this whole part right here happens inside of the mitochondrial matrix. And then the citrulline, there's a citrulline transport. Let me see if I can find the exact name of it. There's a citrulline transporter. I think that's what it's called. It's either urea cycle. Here we go. Yeah, it just says a citrulline transporter. Citrulline is then transported from the mitochondria to the cytosol by citrulline transporter. Okay. Ornithine transcarbamylase catalyzes blank in the urea cycle. This is still just along that exact same line. We're transferring that carbamoyl group to ornithine to form the citrulline. Still talking about this same step that we just talked about above. The amino group nitrogen present in mitochondrial glutamate must be transported to the liver in the form of which of the following. Remember, whenever we talk about transporting it to the liver, the way that it gets to the liver is through the blood. And what's the form that it takes when it travels through the blood?
Alanine.
Alanine. So immediately that's gone. That's gone. We have these two oxaloacetate and alanine. Let me see if that picture has the glutamine in there as well. There it is as well. Blood circulation. So from muscle tissue, it's alanine. From other tissues, it's glutamine. So whenever it says specifically skeletal muscle, answer is alanine. If it says other tissue besides skeletal muscle, then it's glutamine. If it says just tissue in general, pick both of them, alanine and glutamine. The final product of the urea cycle is urea. Identify the site where urea is finally released immediately after its production. So remember I told you within that cycle, remember, so we have all this that happens up here in the mitochondrial matrix, and then the citrulline goes out and the rest of the whole cycle happens. Where's the rest? Where does the rest of the urea cycle happen?
The cytosol?
Yeah, it's in the cytosol. The specific step and where urea is created, can you remember what goes to what? This step right here. Look, it's when our arginine goes to ornithine, and kind of that last step, urea pops off thanks to the arginase enzyme. So here is arginine to ornithine, arginase enzyme, urea pops off in the cytosol. Good. Any questions about that?
Nope.
Yeah. Another one, just draw it out a couple times. This thankfully, compared to the Krebs cycle, this is not nearly as bad. I wouldn't spend as much time focusing on all the little details, like the ATP is here, and this thing is here, and this whatever. I would focus on the main ones, and then secondary to that fumarate comes out between these two. Aspartate is used here, and urea comes out here. That's about as deep personally as I would go, because I feel like that's easier to kind of start to remember. The overall urea biosynthetic pathway takes place only mitochondria in both mitochondria cytosol, endoplasmic reticulum, and cytosol, or only in the cytosol. I mentioned this multiple times, mitochondria and the cytosol, because our first steps happen in the mitochondria, then the rest of it happens in the cytosol. What's the very first step that takes place in the mitochondria?
Alpha ketoglutarate. Oh, wrong thought.
So the alpha ketoglutarate, that kind of stuff, we're going to see with the transport of the glutamate. Hold that picture again. So in order to transport that, our glutamate gets turned into alpha ketoglutarate, or alpha ketoglutarate gets turned back into glutamate in order to pull out and to transport the ammonia to the liver. Our urea cycle happens here in the liver. The production of urea happens in the liver. So our very first step is we get our ammonia gets acted on by our CPS1, the carbamoyl phosphate synthetase, to make the carbamoyl phosphate. I'm going to keep trying to drill that home. Do it as many times as you can. What is the function of arginase in the urea cycle? What does arginase do? Look at this. So arginine, thanks to arginase. What does arginine get turned into again?
Isn't the argininosuccinate one, that one? Isn't that the next step?
That's going to be the step just before. So we have arginosuccinate here. That enzyme is argininosuccinase, is right there. We have an extra thing of fumarate popping out of there. Arginase acts on arginine.
And then the
to
urea
produce
off right there.
ornithine.
Ornithine, that's what's called.
And what comes out right here between arginine and ornithine?
Urea.
Yeah, our urea. So the whole function of the arginase is to create what?
The urea?
Yeah, to form the urea. That's its job. How many nitrogens does urea have?
2.
2, good. And where does this take place again? Where does the urea cycle take place? What organ does it take place in?
The liver?
Good. And where is it getting the aminos from? What other tissues? What other big tissue, main tissue do we talk more about?
Skeletal muscle?
Muscles.
Good. Yeah. And our the glutamate hands off the amino group to what other compound, what other molecule in order to form alanine?
Pyruvate?
Perfect.
Good. Pyruvate. And what does that glutamate turn into when it gives up that amino group?
Alanine.
Oh, so the glutamate, gives the amino group to the pyruvate. Pyruvate turns into alanine, and glutamate turns into alpha ketoglutarate. Yeah, glutamate turns into alpha ketoglutarate, and then when it gets all the way through the blood into the liver, there's alpha ketoglutarate waiting there, and it takes back the amino group from the alanine in order to turn back into glutamate, and then glutamate goes off and does the thing to form urea. Okay. What would be the impact of the presence of high levels of arginine above normal conditions on urea synthesis? So this one, we haven't mentioned this one specifically. We'll think so with high levels of arginine, are we going to want to create more urea or less urea?
More.
Yeah, because if we have high arginine, we're going to want to push the reaction forward. Arginine goes to ornithine thanks to arginase, and it produces the urea. So what is the when we look here, so first of all, we know it will not decrease production. So we look and it inhibits the reaction between silicon and glutamate. No, it's not going to inhibit those kind of reactions. Of these things are N-acetylglutamate. What does N-acetylglutamate do? What's its job?
It acts on the CPS1.
Yeah, and what does that CPS1 do?
Creates the hang on.
It allosterically activates something. You might have said it, and I just didn't hear it.
The NAG.
Yeah, the N-acetyl glutamate is the NAG. And that NAG is going to go into that mitochondria and activate our CPS1. This way, this one is going to be our answer. So it's going to increase the synthesis of N-acetyl glutamate, which is going to drive the reaction of the urea cycle forward and making it happen. Which amino acids are metabolites in the urea cycle? So we're looking to see, okay, which of these are in the urea cycle itself, but are not used as a building block of proteins.
Ornithine and citrulline.
Good. So these are both considered amino acids because they have this same like chemical structure, having the R group, the carboxyl group, and then the amino group. But because they're not used to actually build the proteins, these are just used within the urea cycle. That's like why our very first question was all of the above, because these guys count as amino acids. He likes that question, that first one. I almost guarantee you it's going to be on the test because he wants to check, because technically, like, arginine is like the only one that people think is actually an amino acid when it's all of them. Okay. Which compound is not involved in the biosynthetic pathway of urea production?
Citrate.
Good. Where's citrate from?
Citric acid cycle.
Yeah, from our Krebs cycle or citric acid cycle. I don't think he'll ask this specific question, but if he does, during the urea cycle, during the whole entire cycle, we use 2 ATP in the beginning and 1 more ATP here. So for each cycle, we're going to use 3 ATP to create urea. I don't remember him asking that type of question, but just in case. Which enzyme catalyzes the synthesis of glutamate from glutamine? This one we haven't mentioned or talked about yet. This one is called glutaminase, and that's its job to turn synthesis of glutamate, so it's to take the glutamine and turn it into glutamate. Please don't freeze, computer. There we go. I was just looking at the book before this. It should be in here. I can't find it. We'll see while we're right here. This question is going to come up. So he loves this type of question. So within the urea cycle, he'll ask where each of the nitrogens come from. The first nitrogen of urea comes from the NH4, comes from the ammonia. He can substitute, instead of saying ammonia, he can say from glutamate, technically is where the first one comes from, or from like other amino acids. So the first one comes from ammonia, and the second one comes from the aspartate. So aspartate is here off to the side. This is where our second nitrogen comes from. So first nitrogen from the ammonia way up here at the very beginning, right here. Second nitrogen from aspartate. We'll probably see that question later on in the LINUS questions. Which of the following amino acids can be synthesized from one of the intermediates of the citric acid cycle and participates in removing the toxic ammonium ion from our body as urea?
Alanine.
The answer to this one is glutamate.
Why?
I don't believe because within the citric acid cycle, alpha ketoglutarate, which is very, very similar to glutamate, those two, glutamate's connected to the citric acid cycle. While alanine is technically correct that it does participate, it doesn't have the connection to the citric acid cycle itself.
Even though it comes from the pyruvate from when you with the skeletal muscle and stuff.
Let's dive deeper into the question and see. I don't believe alanine. Let's also look. Oh, I need a new computer so bad. Are you guys still seeing the screen, or is it a black screen for you?
It's black.
Okay, let's give it a sec. I guess we won't be looking it up because the internet makes my computer crash. Yeah, my best guess is that because the alpha ketoglutarate is part of the citric acid cycle, whereas pyruvate technically isn't part of the citric acid cycle. Pyruvate gets turned into acetyl-CoA, and then acetyl-CoA is what enters into the citric acid cycle. That would be my best guess as to
That
why.
makes sense.
As well as it's specific about the amino acids.
That makes sense.
Whether that's 100% correct or just part of the way correct, that's the way that I interpret it and that I understand it. So I'm sure Dr. Sarkar could write a whole book about this one question, about why it is the way it is. But which of the following compounds allosterically activates carbamoyl phosphate synthetase 1 in the urea cycle?
N-acetylglutamate.
Yeah. See, a lot of these questions are pretty similar to one another. Which of the following compounds generated from an intermediate of citric acid cycle incorporates the second nitrogen atom of urea in the urea cycle?
Aspartate.
Yeah, aspartate is the second one. Where does the first one come from?
The ammonia.
Yeah. I believe ammonium ion, if it specifically says ammonia, if you have both options, ammonia versus ammonium, I would pick ammonia. But if ammonia isn't an option and ammonium ion is, then I would pick that one. It's as far as, so let me actually write that in there. First one from ammonia, that isn't a choice. Pick ammonium ion. Part of the fun game of all the beautiful AKAs of science. Okay, which of the following compounds incorporates the second nitrogen atom, or the second second nitrogen atom of urea in urea cycle. This is the exact same question as this, just asked a little bit differently. It's the aspartate. Throwing a little physiology into this. Glutamate as a neurotransmitter in the spinal cord. What does glutamate do? Does it activate or does it inhibit?
It inhibits.
Almost there. What's the other choice?
Activates.
Yeah, so always remember, glutamate
Activates.
activates. Glycine, with an I, inhibits.
Right.
That doesn't go away. You'll see that a couple more times in future courses. Which of the following compounds is required to synthesize citrulline from ornithine?
Is that the carbomoyl phosphatase or phosphate?
Good. And what's the name of the enzyme that helps? Actually, no, never mind. It doesn't give us an enzyme. I'm assuming that there would be one. Maybe not. I don't know. I won't worry about it. Doesn't say an enzyme, so I'm not going to worry about it. Which of the following enzymes in the urea cycle produces a glucogenic compound, which is also an intermediate of the tricarboxylic acid cycle? Which of the following enzymes in the urea cycle produces a glucogenic compound, which is also an intermediate of the Krebs cycle? Let's think about this. Which one?
It's one of the arginos, right?
Yeah, it is.
Yeah, I was gonna say A.
It is. Yeah,
And I
good.
was gonna-
Arginosuccinase. Do you guys remember what the compound is that gets created by the arginosuccinase?
Can't remember.
Are you sure you can't remember?
That and I'm chewing salad.
It's fumarate.
Awesome. Way to go. What does arginase do again?
It acts on the arginine to form the I don't remember that whole word. And the urea.
Ornithine.
Yeah, that
Produces the
ornithine.
urea. Good. The arginosuccinate synthetase. What else is involved with the arginosuccinate synthetase in order to form the arginosuccinate? What else enters here? Give you a hint. We just had 2 questions about it.
Mate.
The aspartate. Aspartate comes in during the arginosuccinate synthetase process to form arginosuccinate. Okay, which of the following enzymes is the rate limiting enzyme in urea biosynthesis? I promise you, this exact question is going to be on the test. I promise it's going to be on there. Rate limiting enzyme. This one. We look at this. Our very, very first step, our mitochondrial carbamoyl phosphate synthetase 1. Because if we never make the carbamoyl phosphate synthetase, we can never form the citrulline from the ornithine to start the whole process in the cytosol. Which enzyme in the urea cycle finally catalyzes the release of urea?
Arginase.
The arginase. A lot of repeat questions here. Which of the following group of chemicals is required to synthesize carbamoyl phosphate in the urea cycle?
It's one of the two with carbon dioxide, if I remember right.
So
Or
carbon
ammonium
dioxide.
ion.
Remember in Biochem 1, we talked about buffer systems.
Bicarbonate.
Right? The bicarbonate buffer system. So if you see carbon dioxide and bicarbonate, technically carbon dioxide turns into bicarbonate within that buffer system. But yeah, so we have right here our bicarbonate, ATP. What was the other one that has in there? And the ammonium ion. There's one other thing in here. There's one other cofactor that's in here as well. Do you guys remember what that cofactor is?
Or hold up.
It's the B6.
B6.
Starts with a P. Can't remember the whole word, but
Dr. Sarkar made a huge deal about it in our small group session the other day. Not to mention he said he was babysitting us, which was quite comical.
It is pyridoxal phosphate. Pyridoxal phosphate.
The active form of pyroxidine, correct?
Yeah.
I did retain
Correct.
something from that day. Or pyrox. He actually did a review session for the blended class today. It was only me, Cheryl, and Kat. Guess what? He didn't post the live video for everybody else because he's tired of people not coming to class. So he sent us the link from the live from your class, Liz, and he sent it to us so we could get the full review where everybody else will just get what he recorded with us today.
Nice. There probably wasn't very many people in there for us today either. I wouldn't know. I wasn't there.
I think he said he had 15 to 20 at least show up for you guys.
That's nice.
Well, here's
People
the problem.
freaking out the test.
That's part of the problem is everybody. He's like, I joked with Kat because we were texting back and forth when we were doing it, and her and I joked. We go, everyone who didn't attend review gets a really, really hard test. Everyone who attended review gets a really easy test with one question. What's your name?
That would be nice. Doesn't work that way though.
Oh, trust me, I've had a few professors do that. Not at Parker, but at Ball State. If you made the effort and attended all the review sessions, the chance of you getting a hard test versus an easy test, because all our professors randomized test, so they had like a bank of like 10,000 questions, and they could sort them by easy to hard, and he'd give all the easy questions to the people who actually applied themselves and studied.
Nice. That's not the way it works here.
Oh, I know.
Okay. Which of the following intermediates of the citric acid cycle can be directly used to produce aspartic acid by a transamination reaction?
Fumarate.
What was that?
Fumarate or succinate? Or am I all wet?
This one is oxaloacetate. This is kind of a... I don't love this question just because this is a lot of like think way back to try one material basically.
Okay.
I don't think, let me see if he has anything in here in the book that specifically says this. Yeah, I don't see anything in here that specifically gives that. So Yeah, just if he has that question, this is one that I'd star in here just to review what the answer is. If you see aspartic acid, it's oxaloacetate. Which of the following intermediates of the citric acid cycle is generated in the urea cycle?
Fumarate.
Fumarate. Which of the following intermediates of the tricarboxylic acid cycle is generated during urea biosynthesis that also produces malate? Again, fumarate. I have a feeling that he's probably going to include a question two like this on the test because he asked it 1,000 times. Which of the following is/are not required to synthesize carbamoyl phosphate in the urea cycle? Answer to this one is going to be citrulline because this comes after.
I was gonna say that, but I talked myself out of it.
Don't do that.
Go with your gut. Let's go with your gut.
Yeah. So we go from our ammonia, CPS1, we put in ATP, the bicarb, 2-ATP technically, HCO3, and then we use our B6 to do that whole process. Speaking of which of the following is a coenzyme required for all transamination reactions? Or pyridoxal phosphate, aka B6. Which of the following is the major nitrogenous excretory product of amino acid metabolism in humans?
Urea.
Correct, urea. Which of the following is/are the major nitrogenous excretory products derived from amino acid metabolism?
Isn't that basically just the same question?
Very similar, just a couple different answer choices because the answer choices are different. Urea and the ammonium ion, aka the ammonia, are both derived from the amino acid metabolism. So technically the correct answer is all of the above on this one. Which yeah, I don't love that double question because these two questions look way too similar in the fact that it has uric acid up here and there. I don't like that. It's probably like the devil's in the details of like the specifics of the questions being like the product of amino acid biometabolism and then like derived from amino acid metabolism. That's probably like a little bit of wordplay specifics, which is I don't like, but which of the following nitrogenous excretory products derived from amino acid metabolism is least toxic and generated in mammals? This one is again going to be urea. He has a specific thing that he mentions about mammals in here that I had highlighted at one point, I believe. Maybe not. Maybe I think I just didn't highlight it, but that's okay. Which of the following reaction products contributes and incorporates the first nitrogen atom in urea? Where does that first nitrogen atom come from?
The NH4 ion.
Good. Where does the second one come from?
Aspartate.
Good. Aspartate is derived from. No, I was going to try and repeat that question earlier. That aspartic acid comes from oxaloacetate, but I don't want to confuse us too much. Which of the following reactions is catalyzed by ornithine transcarbamylase in the urea cycle? We look at this. When is ornithine and carbamoyl together? Was that reaction that's occurring when we go from ornithine to citrulline with the carbamoyl phosphate? So the formation of the citrulline from ornithine, another reactant. I knew there was an enzyme. I don't know why the charts and the graphs, the book didn't show right away. The ornithine transcarbamylase is that ornithine to citrulline. Which of the following statements is not true in reference to the mammalian synthesis of urea? This one will just give the answer and we can talk about why. So this is not true. The process of reproduction is energy producing series of reactions. It is not, because how many ATP total do we use during the full citric acid cycle? We use 3 ATP. Yeah. We use 3 ATP, but we don't produce any NADH, FADH, or anything else like that that would offset the ATP that we use. Which of the following statements is true? So this is actually kind of going back. This is the reason why the glutamate and stuff in skeletal muscle, converts to alanine in order to get through, and then why it turns it back into glucose to send it all the way back up through is because the plasma membrane is impermeable to glutamate. So glutamate can't just pass through all by itself. Which part of the amino acid is oxidatively catabolized to form urea? We can look at these immediately and say, okay, not all of the amino acids have aromatic rings. Not all of them have self-hydryl groups. That's only the MCCs. This is where Tyrone trips you in the ring. So, in the amino acids to form urea is nitrogen. It looks more like an M instead of an N. It is nitrogen. And as our amino groups are our source of nitrogen. That one went pretty quick.
I'm used to the like 50 plus question chapters.
Well, this one was still 43 questions, but a lot of it is kind of repeat. I think that the urea cycle looks a lot more intimidating than it actually is. Most of the question stuff, the only other things that I would say that he might, throw in as questions. He might throw a question in about like pepsin, trypsin, these guys. It's just like, hey, what does this do, or where is it located? Like pepsin is in the stomach and it's used to break down the protein peptides, and all these other ones are going to be in the small intestine, do the same thing. They break down the proteins. This right here, you might ask about this, kind of these steps: transamination, oxidative deamination, transportation of ammonia, and then the reactions catalyzed by the enzymes of the urea cycle. This is just the 4 major pathways of how we go from amino acid to urea. So here we have this is our transamination with the amino acid, converting alpha ketoglutarate, taking it from it, doing all that kind of stuff in the liver. This is just, again, essentially saying the exact same thing with amino acids, converting from glutamate to alpha ketoglutarate to go to wherever it needs to go. The alanine and the glutamine entering here and here as well. The shared transport between the liver and the muscles. And then finally, the actual urea cycle. And then last week we saw questions about nitrogen balance, about having a positive versus a negative nitrogen balance. Positive nitrogen balance just means that you are consuming or eating more nitrogen than you are getting rid of. Negative nitrogen balance means that you are getting rid of more nitrogen than you're taking in. So we want to just be at a kind of an equal level for that. Okay. Real quick. Do this. This was urea cycle. Chapters. Why do I have 2 here? I don't know, but one of them is getting deleted. I don't think I sent you guys these last time, correct?
I don't think so, but I don't remember for sure.
Okay, I'm going to go ahead and drop these into the chat right now. And the test is next week, correct?
Week after.
Not till the week after?
Yeah, because we only have micro next week.
Okay. Chapter 8 is... Are you guys doing chapter 8 this week?
Yes, we are. Or chapter 9
Okay.
this week. Or the 1 carbon atom. Or the one we're in the 1 carbon chapter right now.
The biomolecules for 1 carbon group transfer?
Yes, sir.
Okay, so that chapter 8, so that's this week.
But that's the last chapter on the next exam for us. How about you, Liz?
Chapter 8 is?
Yeah.
Yeah,
Okay.
that's the last one. So this through this week is the only thing that will be on the test, but the test is not until the week
Week
after.
10.
Yeah.
Okay. Okay, good. That puts us in a good position. That'll leave me a little bit of time. I will try this weekend to record, because I don't have this chapter recorded for regular videos. I'll try and record this chapter, and then we'll finish the questions, the chapter 8 questions next week, and then that'll kind of put us at a spot where we've gone through all of the questions for the test.
And you have the one for the urea cycle, you said you have a video for?
Yes, yeah.
Okay.
I just watched it myself earlier today. I will say, it is extremely nice to be able to go back and have those stuff recorded where I can just throw it on rather than having to reread the book for myself and figure things out. Previous me did all the legwork. It's fantastic.
You gotta love it when previous you helps out current you.
Yeah. Now he's leaving me hanging out to dry. Now I actually have to do all this stuff again. So save there. Save. What was that? And then this last one isn't super long either. I think it's 20 something questions. Yeah. 27 questions. So this one won't be horrible. This is a lot more talking about more B vitamin stuff and kind of the function of B vitamins. So sweet. Well, you guys, I'm sure you've had a long week.
I need a large alcoholic beverage and a nap.